OTC stands for ornithine carbamoyl, ornithine trans carbamylase, which is doing the transfer of carbamoyl group from carbamoyl phosphate to ornithine. The so-called formed citrate is transferred back to the cytoplasm via an antiporter. Remember, ornithine and citrulline both transfer in or out via an antiporter. The L-citrulline in cytoplasm react with aspartate. Aspartate donates second nitrogen of urea. These both combine to form argininosuccinate. Argininosuccinate further react to form arginine. Arginine is further degraded by arginase to ornithine and urea. This reaction is catalyzed by enzyme known as argininosuccinate synthetase. Synthetase because there is a formation of argininosuccinate and use of ATP. Remember, here ATP is converted to AMP, not ADP, which is equivalent to 2 moles of ATP. Then argininosuccinate is converted to arginine. In this reaction is catalyzed by enzyme known as argininosuccinate lyase. Argininosuccinate formed is lyase to arginine and fumarate. Fumarate is further hydrolyzed to malate, and malate is further converted into oxaloacetate with the help of enzyme known as malate dehydrogenase, and this oxaloacetate is transaminated to aspartate with the help of amino acid known as glutamate, which is converted to alpha ketoglutarate. Now we move to the theory of urea cycle. Urea is the major disposal form of amino group derived from amino acid and accounts for 90% of nitrogen containing components of urine. Urea structure is NH2 C double bond O and NH2, in which one nitrogen is donated by aspartate and another nitrogen is derived from free ammonia. The nitrogen of aspartate comes from transamination and free ammonia comes from oxidative deamination of glutamate. And carbon and oxygen of urea are derived from bicarbonate ion. Urea is produced by liver and then is transported in blood to the kidney for excretion in urine. Now we move step by step to the reaction of urea cycle. First two reaction takes place in mitochondrial matrix and remaining steps takes place in cytosol. And important point is this, that heme synthesis and gluconeogenesis also involve cytosol and mitochondrial matrix. First reaction of urea cycle is the formation of carbamoyl phosphate from 2 ATP, free ammonia and bicarbonate ion. This leads to the formation of carbamoyl phosphate. This reaction is catalyzed by carbamoyl phosphate synthetase 1, also known as CPS1, and ammonia is incorporated into carbamoyl phosphate is provided by primarily by oxidative deamination. The source of this free ammonia is oxidative deamination of glutamate. And ultimately, the nitrogen atom of this ammonia become one of the nitrogen of urea. CPS1 requires N-acetyl glutamate as an allosteric activator, which is positive activator. And we further move to another node, that is CPS2. CPS2 participate in pyrimidine synthesis, which doesn't require NAG as an activator and uses glutamine as a nitrogen source. The second reaction of urea cycle is citrulline formation. Citrulline formation also takes place in mitochondria, in which L-ornithine reacts with carbamoyl phosphate to form L-citrulline and inorganic phosphate is released. OTC stands for ornithine transcarbamylase. Transfer of carbamoyl portion to ornithine leads to the formation of L-citrulline. As we further discussed about this, ornithine and citrulline move through the antiporter present in mitochondrial matrix, and this movement requires an antiporter via mitochondrial membrane. And also a special point about these two amino acids, that these both are basic amino acid and there are no codons for this amino acid to get incorporate into intercellular proteins. These basic amino acids are not incorporated into cellular proteins because there are no codons for them. Third reaction, argininosuccinate formation. L-citrulline reacts with aspartate with use of ATP to argininosuccinate and release of AMP. The alpha amino group of aspartate provides second nitrogen of urea. ATP is converted to AMP. About this we already discussed that if ATP is converted to ADP, let X amount of energy is released. If this ADP is also lyased to AMP, then also X amount of energy is liberated. So we can write that ATP to AMP energy released is 2x and ATP to ADP energy released is x. And this is the last ATP used in urea cycle. The total ATP used in urea cycle is 2 ATP in formation of carbamoyl phosphate and 1 ATP in formation of argininosuccinate. So total ATP used in urea cycle is 4, because this ATP is converted to AMP. Fourth reaction of urea cycle is argininosuccinate cleavage, in which argininosuccinate is cleaved to fumarate and arginine with the help of enzyme argininosuccinate lyase. Arginine serves as immediate precursor of urea because this arginine is converted to urea and ornithine, which we will see in further reaction. Regulation or fate of malate. Malate can be converted to oxaloacetate with the help of enzyme malate dehydrogenase, or this malate can enter mitochondria using malate aspartate shuttle, where which it re-enters TCA cycle in which it get oxidized to oxaloacetic acid and then to be used in gluconeogenesis. Conversion of fumarate to malate is a hydrolysis type of reaction, and in which malate dehydrogenase react on malate to form oxaloacetate. In this reaction, NADH is formed. This NADH serves as a ATP producer in urea cycle. Thus, urea cycle is linked to TCA cycle and hence it is called as urea bicycle or Clabs-Hanslett cycle, because urea cycle is linked to TCA cycle at which point? Where malate is shuttled to mitochondria back and where it re-enters to TCA cycle. So it is also known as Krebs-Hanslett cycle. We have already discussed that source of ATP in urea cycle is the formation of NADH by malate dehydrogenase enzyme. The last reaction in a urea cycle is the formation of urea by lysis of arginine, in which arginine is lysed to urea and L-ornithine by enzyme known as arginase 1. This reaction is exclusive to liver only, which means arginase 1 is only present in liver, which is responsible to cleave arginine. In kidney, there is formation of arginine, but in kidneys it doesn't last. But there is a presence of arginase 2, which controls arginine availability for nitric oxide synthesis. Nitric oxide is a special product of arginine. Now we will discuss about the fate of urea. The urea formed in liver is diffused to blood. From blood it travels to kidney. From kidney it is excreted out as urine. Some amount of urea also diffuses in intestine, where it is acted upon by urease, which convert urea into carbon dioxide and ammonia. The source of this urease is intestinal bacteria. The ammonia formed in gut is transported to urine, or by hepatic portal system it is transported back to liver, where it is reconverted into urea. Now we discuss about the applied aspect of this hyperammonia. In cases of kidney failure, urea deficient to gut increases because the excretion of ammonia from kidney is hampered. So diffusion of urea is increased in gut, where it is converted to CO2 and ammonia. So ammonia production is increased, which leads to condition known as hyperammonia. Antibiotic treatment. Antibiotics are given in which they reduces the number of intestinal bacteria, which ultimately decreases the concentration of urease. Hence formation of ammonia is decreased and patient can overcome hyperammonia from this. Now we will discuss about regulation of urea cycle. NAG, N-acetyl glutamate, is an essential activator of CPS1, carbonyl phosphate synthetase 1, and as we already discussed that it is a rate limiting step. It increases affinity of CPS1 for ATP. Synthesis of N-acetyl glutamate. Acetyl OA combines with glutamate to form N-acetyl glutamate, and this reaction is positively activated by arginine. An enzyme used is N-acetyl glutamate synthetase. Degradation of N-acetyl glutamate. N-acetyl glutamate is degraded to acetate and glutamate.